pleasure to be here. It's actually the first external seminar that I've done. I was telling a few people before this started. So I started my career at Central or at YVC, and then research career at Central, and then I'm starting the new part of my career here at Central as well. So it's an honor to be back. Thank you, Lucinda and Jason, for inviting me out to give a talk. Oops, that's my chime telling me I've done something wrong. Okay, so I just wanted to go, Jason talked about this briefly, but I wanted to kind of talk about just real quick sort of my path. So you mentioned that host pathogen interactions was sort of what I've done, and it's in fact what I still do. I still study how hosts and microbes interact at mucosal surfaces. So at Central, I studied the frog fungus chytrid and its distribution in central Washington state, as well as its pathophysiology. And this research, that I did at Central, I come to find out in a conversation over a beer at a conference with the faculty at Davis, this research was actually the driving factor for why I was admitted to UC Davis. So it's very important. This was a crucial part of my career, getting this experience working with Jason and Steve Wagner and Jim Johnson. I went on to do my PhD at UC Davis, and there I studied how HIV infection impacted epithelial gene expression. And one of the things that I got involved with there is this project where we looked at how hosts and commensal microbes were interacting during HIV infection, and found that the disruption of those microbial communities drove some of the inflammatory patterns that we observed in the gut during HIV infection. So this got me really interested in the microbiome and the gut microbiota, and how those, or how they might be involved in promoting health in humans. But I realized that I didn't have the sort of bioinformatic or biostatistical expertise that I needed to really be doing this sort of high level work in microbiome science. So I convinced over another beer a new assistant professor at Oregon State University, Tom Sharpton, to take a chance on me. And I spent about 6 years there learning how to analyze microbiomes and design microbiome-based experiments. So the primary focus of my work there was understanding how hosts and microbes interact with environmental stimuli. And after all that, after 11 years, a long road after Central, I landed a job here in the Department of Pathobiology at University of Illinois, where I have still managed to start a lab despite all this COVID business that we've been experiencing for the last year. So it's been a lot of fun doing that. So as I mentioned, I studied as a postdoc host microbiome environment interactions, and I have launched my lab studying the same thing. And I wanted to talk to you a little bit about a couple stories that I developed as a postdoc that I'm continuing as the faculty, and where those stories are headed in the future, and hopefully set up a sequel to this talk maybe 4 or 5 years down the road once I get some good data. So first, I'll give a little bit of background on the topic. So we are exposed as humans to thousands of different environmental exposures, dietary exposures, including macro and micronutrients, commensal microbes, pathogenic microbes, as well as different environmental chemical exposures. And some of these events have an influence on health. But one thing that we've noticed over the past many years studying this is that responses to these types of exposures tend to be highly individualized. So I might not react the same way to a COVID infection, say, as you do, or I might not react the same way to consumption of high amounts of fiber in my diet as you would, right? So although there have been many genetic factors that have been identified that explain away some of this variance, much of the variance is actually still unexplained, which has led to this hypothesis that potentially some of these variants in response to these environmental exposures may be driven by the millions and actually trillions of microbes that inhabit every surface of our body. And so these microbes that inhabit not just our gut, but our skin, our mouths, our nose, everywhere, form beneficial symbioses where they prime the immune system, respond to pathogens, they detoxify chemical exposures, and they help digest undigestible components of our diet. Right. And an important observation in microbiome science about 20 years ago was the disruptions of these communities oftentimes associates with the development or severity of diseases. Right. So it's become very important for us to understand how the microbiome executes these functions, what kinds of exposures could perturb the microbiome and disrupt these functions in order for us to use the microbiome potentially as some sort of biotherapeutic in the future. So as I mentioned, the microbiome interacts extensively with the environmental exposures that we experience, which has led to this hypothesis that these microbes may actually be contributing to the differential responses to exposures. So I mentioned I'm going to talk to you about two stories today. The first one I'm going to talk about is one of my favorites, something that I started very early as a postdoc, working as a postdoc at Oregon State, and that's defining how the gut microbiota interact with intestinal helminth parasites. Right, so intestinal helminthic infection is a widespread health problem. So this map shows the level of, or the proportion of children that require chemotherapeutic preventative measures to stop infection with intestinal helminths throughout the world. This map was put up with the World Health Organization. And you can see a large portion of the world, over 2/3 of the children in those countries require these chemotherapeutic interventions. So currently over 1.5 billion people are infected with these intestinal helminths on a yearly basis. They have significant health and economic impact. So while they rarely will kill a person that's infected with an intestinal helminth, they oftentimes don't make you feel so good. They have other morbidities that associate with decreased productivity, as well as decreased healthy lifespan in these individuals that become infected. And so concerningly, there's also growing drug resistance. So we have a number of anthelminthic compounds that are directed against these parasites, but the rate at which we're producing new anthelminthic compounds is being outpaced by the rate at which anthelminthic resistance is increasing. Right, so we need new strategies that can either prevent infection or treat these infections after they become established. And so actually so of course we as microbiome people think, well, of course it's the microbiome, we can do anything with that, right? So it's actually not totally crazy, because because microbes for a long time have been sort of the workhorses for developing these new drugs. As a matter of fact, in 2015, Dr. Campbell and Amura won the Nobel Prize in Physiology and Medicine for the discovery of ivermectins, which are a class of compounds that are produced by Streptomyces bacteria that are strongly antihelminthic and well tolerated at high doses in individuals. So these drugs really have been transformative, and they're produced by microbes, in this case, a soil microbe. There are other microbes that also produce these anti-parasitic compounds, namely Pseudomonas and Bacillus species produce a lot of these compounds. So we thought potentially we could be using these normal microbiota as preventatives because of this ability to produce these compounds. On the flip side of this, microbes can also enhance helminthic infection. So in some cases, especially in the case of Trichuris murus and other Trichuris species, the worms actually need bacteria to signal that they've reached somewhere where hatching is appropriate. So in this case, a protein in the sort of attachment pilus or fimbriae in a bacteria attaches to the outer a portion of the parasite egg, which signals to the parasite that is present in a gut environment. The parasite hatches and initiates infection. And so when you eliminate those bacteria in the gut, you get a much lighter infectious burden. So there's a double-edged sword here. We have to be careful if we want to use the microbiome, because we could end up enhancing infection instead of preventing it. So the primary thought that we had going into, or the primary motivation for the work that I'm about to describe, was trying to understand whether or not we could use the microbiota to prevent or treat infection. And so to get at this question, we developed a model of zebrafish, using this common zebrafish pathogen, Pseudocapillary tomentosa. So this is a SEM image of two worms actually sort of snaking through the epithelial tissue. They're kind of gnarly looking, pockmarked, and particularly scary in this false color blue. But anyway, these pathogen, or these worms, like I mentioned, are common zebrafish colony pathogens. So you can get a colony that becomes infected, and over a long period of time, because the symptoms of infection are so slight in many individuals, you have most of your colony get infected, and then you have to end up culling all the fish in order to establish a pathogen-free colony again. So this is obviously a huge problem in fish facilities, but it's also very similar to mammalian whipworms, both phylogenetically and in terms of the symptomology. I mentioned that you don't oftentimes get very ill with these worms. You have the majority of fish that maybe lose a little bit of weight, but all in all, they look pretty okay until about 2 or 3 months past the initiation of infection, and when they start to get really sick. So the thing that really kind of pushed us over, that really kind of sold us on this model, was actually that we noticed when we did some preliminary work that when we tried to infect a fish with a standard number of eggs, we got these skewed burden distributions, wherein, here I'm showing you a plot. On the x-axis, you have the total number of worms that we can observe using a wet mount in the gut versus the total number of individuals that have that particular count. So this is just a histogram. And what we notice is that a large or a small number of individuals actually had bore the majority of the burden. So very few individuals were highly infected, where many individuals had light infections. These worms also had extensive interactions with the gut microbiota. We took several SEM images and showed that there were, in many cases, microbes directly in association with the parasite. And of course, the parasite is initially snaking through this milieu of gut microbes that are inhabiting the gut in the first place, right? So we reason that this skewed burden distribution could potentially be due to differences in microbiota abundance in the gut. So we asked the question, does microbial abundance associate with parasite success? In this case, the measure of parasite success that we were particularly interested in was parasite burden. So we exposed a large cohort of adult zebrafish to a standard amount of 200 eggs per fish. And we allowed them to develop infection, and we serially sampled these individuals over several time points. And each time point, we would collect feces from the animal, then euthanize the animal that gave the sample, and do intestinal worm counts. Unfortunately, at the time, there was not a passive parasite abundance quantification measure that we had access to. So we measured burden. We also measured the degree of tissue damage, or the histopathology, that was present in each worm, to determine whether or not the microbiome could also be involved in some of the pathological damage that we see in infection as well. And we measured the abundance of microbes using 16S ribosomal RNA amplicon sequencing. So what we found was consistent with the work of our previous work of our collaborators, was that at around day 30 post-infection, 30 to 40 post-infection, we saw the peak of parasite burden. But this peak didn't actually correspond to peak histopathological damage. As a matter of fact, we didn't see much in pathological damage until much later in the infection. So in this image, I'll just use a laser pointer here real quick, if I can do it. There we go. In this image, you see a typical intestinal villus of a fish. I'm outlining the epithelial cells. And you see that all this is intact. Underneath these epithelial cells is the lamina propria. It's a nice thin layer. And in this group, or in this particular organism, you have a very thin layer of muscle underneath this. So you don't see any infiltrate here. So this is a very healthy gut. You also see a number of goblet cells, which produce mucin, which is, produces the mucus in the gut, which is very, very good for normal digestion and intestinal transit. At day 21, we just start to see some histopathological damage. And you can see here some apoptotic bodies, so dying cells, here, here, and here, as well as a pseudocapillaria worm present here eating the epithelium. But I think you can appreciate later on in infection, the damage becomes much, much more severe. So here you see an ablation of epithelium in areas, a complete blunting of epithelium in other areas. You've lost that villus structure, which means you've probably lost a large proportion of your absorptive capacity. And you also see some of these dark staining lamina propria lymphocyte infiltrates. So you have a proliferation or an infiltration of immune cells into the tissue. So this is a very sick fish here. But this doesn't occur until very late in infection, about 2.5, 3 months into infection, well past the peak parasite burden. So there's decoupling there. So next we wanted to know whether or not any of this tissue damage or burden corresponded to sort of total community diversity. So in order to sort of compress the microbiome into a singular point, we use non-metric multidimensional scaling. And so what this does, as I mentioned, it kind of pushes everything down into 2 dimensions, which allows us to visualize the total community diversity in a single plot. And so each point is a fish's microbiome. The distance between the points is proportional to the dissimilarity or the similarity of these microbial communities. So when you have 2 points very close together, they're very similar in composition. When you have 2 points very far apart, you have a very distinct microbiome of these individuals. So the points are labeled by unexposed individuals in triangles, exposed individuals in circles, and they're colored by the degree of intestinal worm burden. So what we noticed was that while there wasn't great separation in these groups in these 2 dimensions, we did notice there's this skew to one side of the plot where the majority of individuals that had high burden tended to be over here in the left side negative coordinates. So we asked whether or not this diversity or this dimension of diversity correlated with parasite burden. And indeed, we found that it did using a Kindle correlation analysis. So microbiome diversity is stratified by parasite burden. So we can do the same sort of analysis using histopathology. So we can score the tissue damage on a scale of 1 to 6. It's actually a combination of multiple measures. And we can plot it the same way. And we saw the same sort of trend, this sort of skew to the left-hand side of the plot, which again was associated, negatively associated with histopathology. Right, so from that, it convinced us that there was something to this, that the microbiome was in some way associated with these 2 factors, histopathology and parasite burden. So next, to dive a little deeper, we wanted to look at individual taxa and how they associated with burden and histopathology. And I'll start by saying I don't expect you to be able to read this tiny writing. I just put this up there as an example of the number of taxa that has had associations with these factors. And I wanted to call out just a handful of examples here. So the first thing that we notice is that several taxa that typically are associated with infections in zebrafish, including mycoplasma and spiroplasma, were positively associated with parasite burden. The taxa that we know to be typically associated with zebrafish, healthy zebrafish gut, including Plesiomonas, Cetobacterium, and Vauxella, are all negatively associated with burden. Now, one possible interpretation of this data is that the microbiome, there's actually components of the microbiota that are antagonizing parasite success in the gut. Of course, there are other interpretations as well. We can also do the same. We can also look at histopathology and find that a number of highly abundant, common in healthy fish taxa are negatively associated with the histopathological damage. And again, one possible interpretation here is that these taxa might mitigate some of the inflammatory damage by producing anti-inflammatory compounds, which we know to happen in other contexts, such as inflammatory disorders in humans. So I'll get back to this in a minute, but this kind of brought up these really strong signals that we observed in the gut, brought up this question for us, is can we actually use the microbiome to predict the exposure status of an animal? And so what this could allow us to do, because oftentimes these tests for these organisms in the field are costly and time consuming, it would potentially allow us to rapidly screen individuals and identify those that are at most need for either prophylactic treatment in communities where these parasites are endemic, or treatment for the parasite itself, right? Of course, assuming that held with with other helminth infections. So what we did was we built a random forest classifier, which essentially asks, can we determine which individuals are exposed to the parasite based off of the composition of the microbiome? And so here we're really hoping to get something like a 80% accuracy would be a really great classifier. And when we put this into the algorithm, what we got out was this incredibly accurate model. So we actually only had 10% error for our model and an incredibly high level of sensitivity and specificity. So lots of true positives, lots of true negatives. Right. One thing that I've learned working at Central and also working as a PhD is when you see data that looks really good, you've often done something wrong. So I went back and double checked my work a couple different times and still wasn't satisfied with this. So what we did was we took a separate population of fish and exposed them to the parasite. And we asked the question, can we use our model that we built with this first cohort to predict the outcome in the second cohort? So if you can, it means that your model probably isn't overfit using this machine learning technique. And so the result of that was, again, really, really excellent. We had 97% accuracy in classification. So I finally reluctantly concluded that this data was good and that we had a really accurate classifier, which suggests that the microbiome can predict intestinal helminth infection, at least in zebrafish. So we can look at the important factors here. So the most important taxa, and again, you see some of these taxa that I talked about in the previous slides, the Palamonas, Plesiomonas, Vauxella, and Shigella, et cetera. So these are all really, really important taxa in determining whether or not an individual has been exposed. Right. So after we got this data back, we really started talking about, what it could possibly mean. And what we all concluded was that, we have these associations. And like I said, one interpretation was that the microbiome is producing something or interacting with the parasite in some way that negatively affects the parasite success. And other taxa are positively affecting that success. So, but what we all agreed on was that the flip side of that was that there was an inflammatory environment, and these microbes are just disrupted by that inflammatory environment and pushed out, right? So one thing that we wanted to look at is there any evidence that we can find in the zebrafish gut microbiome that would suggest that these microbes could produce natural products that affect parasite burden or pathological outcomes. So we couldn't do this with the data we had. We only had 16S sample con sequencing data, which is very good at telling you who's there, but doesn't tell you what sorts of genes that they're encoding. So next we went in and we looked at the whole, we looked at the total genomic content of the zebrafish microbiome in a subset of animals. And so to do this, you put a postdoc in a room with some fish, and you get really good at doing the fish poop dance to try to get these fish to defecate even when they're kind of stubborn. After you get all that poop out, you extract DNA, you chop it up into these tiny pieces and sequence it. Then you put it all back together again with a special assembler. And from there, you can find genes or biosynthetic gene clusters that may be encoding the genes of interest. And you can annotate these genes in a number of different frameworks. In this case, we use the Anti-SMASH database to annotate this DNA that we were sequencing. And so what we found was that zebrafish actually, the zebrafish microbiome, even the small amount of sequencing that we did, uncovered hundreds of different natural products. And this is not terribly unexpected, but it did show that we had things like non-ribosomal peptidase synthetases, which are antimicrobial, cytostatic, or immunomodulatory in nature. So potentially could be affecting the immune environment in the zebrafish. We also saw bacteriocins, which can act to dislodge other microbes. There's a large number of those. So this could possibly be compounds that are involved in disrupting microbial communities during infection. But we did find a number of polyketide synthetazers. And when we looked to see what the closest relatives of these polyketide synthetases were, at least 2 of these biosynthetic gene clusters had genes that were similar to the genes that encode the pathway which produces avermectin, which, if you recall, is the anthelmintic compound that, or class of compounds that won the Nobel Prize in 2015, or half of it at least. So indeed, we do have these the potential, at least, to produce these things in the zebrafish gut. So where do we go from here? And we thought about this a lot. And one of the things that I came up with is well, if we ever want to do any downstream experimentation, we need these bacteria. We need to culture them out, because we're just looking at sort of omics data, and that's not ever going to really tell us whether or not there's that causal relationship there. And so this is where I work with undergrads quite a bit as a postdoc, and now in my own lab, where we have a system where we routinely isolate fish feces. So now I can put them in a room with the fish and let them watch the fish poop. They come back, they isolate microbes, and they amplify marker genes. And that allows us to go back to that shotgun metagenomic data that we have and ask the question, is this taxa that we've isolated, or taxon that we've isolated, is it associate, or is it the same taxa as something we might be interested in, a bacteria that might be associated, negatively associated, positively associated with with parasite burden or histopathology. If it is, then we can go through the process of sequencing its genome and looking to see what sort of natural products might be encoded there of interest. So far, we have gotten through over 100 different isolates from 20 different genera, and we're just now starting, as I ramp up my lab, to look to see if we can find any particular compounds of interest. Okay, so hopefully what I've convinced you of is that there's some association between the microbiota and parasite burden or parasite-associated tissue damage, and that these might be mediated by natural products produced by these bacteria. At least there's the potential for that, right? And if we are able to do that, if we're able to identify those compounds, isolate them, we may be able to identify compounds that we utilize for parasite control. Okay, so the ongoing work, I kind of mentioned some of the cultural work that we're doing, but we're really interested in getting at the molecular mechanisms that underpin some of these interactions, both positive and negative. We're really interested in how microbiome operation changes, so the actual genes that are being expressed in the microbiome, how they change during parasite infection, and how this changes the production of whatever natural products may be being produced by these bacteria. So now for something totally unrelated, I'll move on to the second part of the talk. And so this is very different work. So one of the things that I was introduced to very early in my postdoc was a collaborator's work investigating how micronutrient deficiencies impact health. And in talking with this collaborator, we really started to wonder whether or not, micronutrients could be influencing the operation of the microbiome, and if that change in operation had anything to do with micronutrient deficiency associated health defects. Right. So when I went to the literature, and the first thing you see when you do microbiome literature search is probably something to do with diet, because we've done a lot of work in my microbiome world of understanding how diet changes the microbiome. And so we know that you can rapidly alter microbiome structure and function by simply giving someone a high-fat diet. And then if you give them a bowl of lentils a couple days later, you can switch the microbiome right back. So it's a very plastic situation when you're modulating the microbiome with diet. And we know a whole heck of a lot about how things like fat, protein, fiber, and carbohydrates influence microbiome structure and functioning. Right? But there's far less known about how micronutrients impact the microbiome operation. There's been a few studies that show that micronutrient deficiencies can disrupt just sort of the structure of the microbiome, but not a lot has been done with regard to looking at the sorts of gene abundance differences in those communities. Right, and so for this work, we decided to focus on zinc for a number of reasons, and one of which is that it's one of the most prevalent and important micronutrient deficiencies that are present around the world. So over 2 billion people are at risk of zinc deficiency. You see a number of countries, over 25% of individuals are at high risk. Now, zinc deficiency reduces healthy lifespan in individuals. A cumulative total of over 28 million years of healthy lifespan are lost in a year. And this is because hundreds of proteins require zinc for adequate functioning. So if you take zinc away, you get defects in cognition, in wound repair, in DNA damage repair, and immune activation. So you have systemic level effects of zinc deficiency, which are really obviously very bad for the host. But on the other hand, you also have the microbiome, which also requires zinc. And since we don't really know genomically what's going on with a microbiome that's zinc deficient or zinc restricted. So we asked this question: does zinc deficiency disrupt microbiome operation or its interactions with the host? So again, we use zebrafish to answer this question. We had individuals fed one of 3 different diets: a standard chow diet, a low zinc diet, and a control zinc diet. So these 2 diets at the top, number 1 and number 2, are purified diets. So they're made from egg whites. So we can precisely control the amount of zinc in these diets and how much the animals are getting. So we exposed them to this diet for a period of about 8 months, and then collected feces and euthanized the fish. Right. And so from this, we did shotgun metagenomic sequencing from the fecal contents. We also paired these data with gut transcriptomics data. So we conducted RNA-seq on the whole intestine. This allowed us to look at not just what's going on with bacteria, but simultaneously what's going on in the host and the gut. And then we paired these data together to try to identify interactions between the 2 different systems we were working with, the microbiota and the host. So again, what we did here for whole genome shotgun metagenomic sequencing is we extracted DNA, we sequenced this DNA, assembled it, and then we could map all these reads back onto this assembled and annotated gene framework and count those genes up to look at different gene abundances across the different fish in different diets. So sort of a high-level summary of what we found here was that zinc deficiency, so low zinc diets, associate with a depletion of pathways in zebrafish microbiota that are responsible for detoxification of chemicals. So these are potentially things like antibiotics or other drugs or other chemicals. Right? We also saw a decrease in beneficial interactions. So here we saw a depletion of things involved with bile acid biosynthesis, short chain fatty acid biosynthesis, which is a nutrient that intestinal epithelial cells can use, as well as vitamin biosynthesis. So we have these sort of benefits that the microbiota may be providing the host, and they're going away in these low zinc diets. We also have this increase in many different types of antibiotic production pathways. And this is typically a response that we see when bacteria are exposed to stress. Oftentimes a bacteria can produce a compound, an antibacterial compound that kills other bacteria around it so that it can utilize the resources where others cannot. We also saw an increase in components of cell wall biosynthesis. So altogether, what we saw was an increasingly antagonistic environment that lacked some of the beneficial functions that the microbiota might be performing for the host. In the gut, what we found was interesting as well. So this is a log2. So this is a plot of all the gene expression that we observed in the gut. On the x-axis, you have log2 fold change. On the y-axis, you have negative log 10 p-value. So as you go away from the center, you have things that are changing more. And as you go up, you have things that are more significant. And what we found was that there is an increase in genes involved with immune stimulation, and particularly innate immune cell stimulation in the gut, as well as genes involved with wound repair or wound healing, DNA repair, and importantly, zinc uptake, this is kind of a nice positive control. When you starve an animal for zinc, they increase the receptor or the channel that's going to pull zinc in. We also saw a decrease in gene expression involved in immunosuppression, as well as a decrease in zinc antiporters. So you're stopping zinc from flowing out of the cell. Again, it's another sort of nice positive control. This is exactly what we expected to see happen. So we know these individuals are low in zinc because of that. And finally, we wanted to know whether or not any of the changes in gene expression in the host were correlated with changes in bacterial gene abundance in the microbiome or the metagenome. So this is a very simple plot of the method. All you really do is just to make this correlation network here. So you correlate the intestinal gene 1 with the bacterial pathway abundance 1 over here. And then you construct a bipartite network from those correlations and use community detection analysis to identify highly connected, correlated subgraphs in the structure. So what you get is a very pretty looking hairball. And inside this hairball, you'll see circles and squares. The circles are microbial pathways, the squares are host gene expressions, and we're coloring these individual points based on the subgraph that they belong to. And so a subgraph is just something that's very densely connected with more so to itself than to other components in the network. And these are frequently thought of as sort of subunits or pathways in a larger network context. And so we can dig into these a little bit more. And what we found was when we started looking at the individual components of these subgraphs was that there are a number of these pathways that were disrupted in zebrafish that were fed low zinc that were correlating with genes that are involved in immune response. So potentially these disruptions in zebrafish gene abundance were either caused by this inflammatory immune environment or were causative in this inflammatory immune environment. So this is data that's all been published in bioRxiv. You can check it out. We've also sort of expanded this analysis in a little bit more, and hopefully in the next year or so we'll have the paper finally done. But getting back to one of these findings that we saw on the heat map that was really interesting to me was the disruption of or depletion of genes involved in detoxification of xenobiotic compounds. Because it raised this question, do multiple exposures, so dietary exposure and a subsequent chemical exposure, do they yield synergistic effects? Is the microbiome more sensitive to a second hit by a toxicant if you expose it to low zinc first? And so it turns out that individuals that are at high risk for zinc deficiency also live in regions that tend to be high risk for arsenic exposure. And I'm sure that many of you are aware that arsenic is a major toxicant of global concern that exhibits severe health defects, including death in very acute exposure situations. But this comorbidity with zinc deficiency risk raised the question, a natural question in our mind was, well, since we have this overlap, can we just look to see whether or not a zinc deficient individual and their microbiome are more susceptible to a subsequent exposure to arsenic? And so for this experiment, we actually used mice because our collaborators had to develop a mouse model for arsenic exposure. And we exposed these different animal cohorts to increasing concentrations of arsenic. So I'd say that these are physiologically relevant concentrations of arsenic between 0 and 500 parts per billion. These animals were fed either a zinc adequate or zinc deficient diet. And we allowed them to be on diet for a period of 6 weeks. And then we looked at the microbiome composition to determine if there was an effect. And so what we found is that the microbiomes of individuals fed zinc adequate diets were not terribly responsive to these physiologically relevant concentrations of arsenic that we exposed them to. But when we looked at the individuals that were zinc deficient, you began to see some separation in community diversity here. And when we run a test of association between the concentration of arsenic and microbial community diversity, we found that there was indeed a significant association here between the two. So the microbiome is actually more sensitive at a community level to arsenic exposure than is a microbiome that's exposed to a zinc adequate diet. And so one of the last slides here, we can go in and look at what sort of taxa are being impacted by arsenic. And while there wasn't a community level effect, there were several taxa that did, in fact, associate with arsenic concentration, as well as zinc deficiency. But several taxa that were very important for physiological health in mice were impacted synergistically. So when we look for an interaction effect here in the model, we see these synergistic interactions. And so, some of these taxa like Lactospiraceae, S24/7, and Archimantia all have sort of this commensal anti-inflammatory, health-promoting phenotype. So the disruption or the synergistic interaction suggests that zinc deficiency is negatively impacting the response to arsenic exposure. Okay, so what we found was in this part of the talk was that there was, when we expose animals to zinc deficient diet, there was increased antagonistic interactions, which we hypothesized moving forward that potentially those could be related to some of the physiological outcomes associated with zinc deficiency. They were depleted in xenobiotic metabolism pathways, which potentially is the reason why these microbiomes are more sensitive to arsenic exposure. So moving forward, we're really interested in my lab in understanding how zinc restriction impacts arsenic metabolism. We know that microbes can metabolize toxic forms of arsenic to less toxic forms of arsenic. We want to know how those pathways are impacted. We also want to know how much arsenic is metabolized by these compounds or by these microbes. Is it a little bit? Is it a lot? There's not a lot known in that area. And whether or not these differential responses to arsenic influence host responses as well. Okay, and with that, I will sum up so we have, or thank some people so we have time for questions. I'd like to thank all the collaborators at Oregon State University, my PI there, Tom Sharpton, as well as some of the undergrads that I've worked with, the Ho lab that was instrumental in the zinc deficiency and arsenic exposure work, Dr. Robin Tangway's lab, who was where basically all the fish work was conducted for the zinc deficiency, Dr. Michael Kent, who is our collaborator, as well as Dr. Martins in Brazil, who collaborated on the parasite work. And then, of course, all the people that kept the lights on throughout the process, and the people that are now foolishly paying me a salary to do all sorts of fun research.